Hello, hello, hello. All right, can people hear me?
I can hear you, sir.
Awesome. All right. We only got a couple so far. Hopefully some more people will join in a little bit. So just to give the two of you a little run around of what we have here. So ideally, if you can mute your audio, unless you need to ask a question, it just stops background noise and stuff from creeping in and affecting things. At the bottom of your Zoom thing, there's a chat bar. So you can say questions if you have stuff that occurs to you, or you can send me stuff privately if you don't want to share it with the rest of the class. What else? Hey, Michaela. I'm going to share my, where are we at? We can put this up first. So hopefully you should be able to see the PowerPoint up now. So we can use that as a tool to go through it if you have questions, you know, anything like that. And where'd the chat go again? Still working my way around this. Where's the chat gone? Can someone write something in the chat bar? Ah, there we go. Thank you. For some reason, some things are hidden when I'm sharing my screen, so it's a little hard to figure everything out. Okay, so basically, this is how we're going to be doing stuff for the rest of the semester. Hopefully, you've also had a chance to go through the PowerPoints. So the two that have been posted and annotated so far are the chromosome structure and dynamics one, which is what I've got open here, and the transposons. Let's see, here you go. The transposons PowerPoint as well. So those audio recordings are up and accessible. Generally speaking, I would suggest you cover those at least once, if not a couple of times before one of these sessions, because really the best use of this time would be going through any questions you have, right? Because obviously we don't have that ability to do so during a lecture. And so if possible, I'd rather avoid just going through material, you know, as if this were a lecture during these sessions, and actually use this time to clarify any questions, you know, increase your understanding and so on and so forth. Does that make sense? Cool. Okay, so questions. You can, there's a, no, don't want that. Oh, hello, Michael. And some more people connecting too. Awesome.
Hello.
Hello. Okay. Oh, there it is. That's how it works. Sorry, still trying to figure out how to use this. What did I want to do? Ah, I've forgotten what I want to do. Anyway, so there we go. Let's go back to this. So questions. All right, so for those that are joining, oh yeah, that's what I want to do. I think I don't have it here because I'm the facilitator, but there should be a in that toolbar at the bottom, like a raise your hand button or something like that. If you want to ask a question, you can hit that. You can just unmute your audio and shout it out. That's totally fine too. Or you can write it in the chat box as well. Both the chat box and this video are going to be uploaded to Blackboard after class, so you'll have a record of them there if you want to go back and check stuff. Okay, so shall we start? No time like the present, as they say. Who's got any questions?
I have a question about what a karyotype is. Is it just a karyotype is like all the chromosomes that consist of like that organism's genome? Or
Yes, and so just pulled up a nice picture. This here, and there's actually a similar one on the starting page or slide. This is a karyotype. So the distinction between a karyotype and your entire genetic complement is that because you're diploid, you have 2 of every chromosome, right? Does that make sense? And so, but because that's essentially like a duplicated set, right? Because you have 2 of each, it's not really like the simplest kind of form of your genetic information, right? And so that is your karyotype. So your karyotype is what chromosomes you have, and it looks like this, right? So it's a very kind of stereotypical, let's see where it actually comes from. See if you can see this. Yeah, so it's the number and visual appearance of chromosomes in the cell nuclear organism. Typically, it's referred to as the entire genetic complement, right, of that organism. And in general terms, I usually refer to it as what chromosomes you have, right? So it's both a visual thing, which is what this picture shows, and it also kind of refers to as that's what you got, right? I don't know. Does that make sense? It's kind of like an old-fashioned word in a sense, right? Really very much came from when we used to look at DNA like visually, microscopically, rather than actually work with it at like a molecular basis. So it's quite an old-fashioned term.
Okay, thank you.
No worries. Okay, next question. Keep me loaded. Oh, there's my reminder for genetics. Dismiss. Get rid of that. This is going to have to be a very participatory kind of deal, because otherwise it's going to be all kinds of awkward. I'm just going to sit here like staring at the wall or something.
I had a question on that same slide that you were at. Like the example you gave was that chromosome 22 has 23 genes per megabase. Like, how did they come up with those numbers?
Oh,
or
okay.
Is that just given?
That must be Rose, right?
Yes.
Hi. So if you could, everybody, if you could just shout out your name as you ask a question, because I don't have everybody up in front of me, because I have my PowerPoint up. So that comes from the human genome sequencing data. And so because we have an entire map of the human genome, we're able to map every single gene, at least as far as we understand it as being a gene, pardon me, onto individual chromosomes. And so those chromosomes are actually, in kind of molecular terms, you can think of them as kind of like scaffolds, or kind of ladders on which you can place stuff. And so as the genome is built, the whole genome sequence, and we do this for novel genome sequencing projects too, you use that kind of scaffolding, which is like the old mapping data, the kind of stuff that you were doing in genetics lab with crosses, linkage experiments. And then you stick in the new bits of sequence until you build up the entire chromosome. And so we know exactly, at least based on bioinformatics, how many genes are on every single chromosome and where they are. That's where like gene density maps come from over the chromosome as well. And so we know where all of those things are. And so that's where those numbers come from. Obviously, those genes aren't all the same size. They vary hugely from very small to absolutely massive. And they're not equally distributed over that chromosome either. Let's go back down here. Can you all see the PowerPoint that I'm going through?
Yes.
Yeah, okay, awesome.
Yes.
So typically, this is more about chromatin, but it kind of applies to gene density too. There's very few genes at the ends of the chromosome. I mean, technically in the telomeres, there are no genes at all, right? Because it's just a buffer. And there are relatively few genes in the center, near the centromere. This is much more of a kind of structural deal. Remember too, this is a chromosome in cell division, right? So it's condensed down, doesn't actually look like this in normal interphase. Also, obviously, this is one sister chromatid on one side. This is another sister chromatid on the other side, right? So this is a kind of cartoon of a chromosome in a very specific context, right? In anaphase, let's say, or metaphase. That said, most of the genes are present in the arms, right? So end, center, it's a centromere. The arms are this one and this one. They're called P and Q for petite, and I don't know what Q stands for. And most of the genes are found in the arms where the euchromatin is. And obviously, because most of the genes are there, you'd expect that DNA to be mostly euchromatin, right? As opposed to heterochromatin at the end or the center. So it's a very heterogeneous distribution of genes, right? Both between chromosomes, which you can see on this particular diagram, right? And there's really no rhyme or reason by size and number of genes, right? You can find, let's see if we can find a good example. Right, this tiny one here, 22, has way more genes than a chromosome almost twice as large as number 18 over here, right? So, and we don't really know where that variability comes from. Most likely it's a result of chromosome duplication in our, in our history, many common ancestors ago, that produced this chromosome count. Because typically speaking, and though it's not a hard and fast rule, the higher the order of organisms, so if you take mammals as being more complex and evolved than, say, salamanders, right, or nematode worms or fruit flies, the more the higher the order of animals, the more chromosomes we have. Doesn't mean that we have more genes, right? Because that goes back to, oh, where is it? This chat thing's in the way. Stick that down. Ba-dum. Don't have a good example here, but these two organisms here have approximately the same number of genes, right? So we're looking at a fish and a, I don't know what that is, kind of cross between a snake and a salamander. If you look at the genome of C. elegans, it has, last count, about 21,000 genes, give or take. So this is a soil worm, which is about a millimeter long, 970 cells, much simpler than us. Our genome has about, actually you could add it up from this this slide, but it has about 22,000 genes in it, as far as we're aware, right? So, but C. elegans has, gosh, yeah, I should know this better than this, 6 chromosomes. Yeah, that's right, including the sex chromosome. We have 23 chromosomes, including the sex chromosome, right? So there's not really any correlation between organismal complexity in terms of you know, features like legs, arms, you know, behaviors, life cycle, so on and so forth, and gene number or genome size, right? But there does seem to be a correlation between complexity and chromosome number, right? So essentially, these chromosomes that you can see here are a result of you know, many tens of millions of years of evolution and duplication and new functions and so on and so forth, which hasn't happened with simpler organisms. I'm not going to call them less evolved because that's a little bit kind of judgmental in a sense, but certainly simpler organisms, right? They have the same number of genes, but they have typically far fewer chromosomes. So anyway, that was probably a long-winded answer to your question. All right, next question. I'm trying to see, who do we have here today? We don't have as many people as I expected. Well, all the load's going to be on you lot. So I can ask people questions. Should I do that? It's kind of like bullying your class. Oh, Michael's stepping up. Come on, Michael, ask me a question.
So I couldn't really catch this in the recording, but you said that there are approximately 15 to 22,000 genes for eukaryotes, but that was the end of your statement. I couldn't quite understand the beginning of the statement.
So I think that was really when I was talking about this, right? The C-value paradox.
Yes.
Right. So, and that's probably even better explained here. So typically, and this is obviously it's a pretty wide margin, but as far as I'm aware, right, and obviously I'm not like an expert in comparative genomics by any stretch, but I don't know of any organism that has fewer genes than 15,000, give or take. Maybe obligate parasitic protists, like not malaria, because that's I mean, it is a parasitic protist, Yeah, so maybe organisms that kind of rely on a host, right? So they need fewer genes to do less things because they use those products from their host, right? So for example, you know, we need to make glucose from complex carbohydrates, right? If you're a blood parasite, you don't, because that's done for you by the host. So you don't need those genes, right? So at the lowest end, like the simplest, most basic eukaryotes, around 15,000 genes. The most complex well, actually, it's not even due to complexity. The highest number of genes that I'm aware of is perhaps 24,000 genes, give or take. Right, and so these are individual, you know, bits of information for encoding a protein, right? And so if you look at this, right, we go from, and I'm not 100% down on what all these different things are because I'm not a taxonomist, but you can go from stuff that's super simple in terms of physiology, right? So single cell, like a malaria parasite, actually it's really cool. Malaria is really interesting. That's not a good example. Let's say one of those funky pond things. Shoot, what are they called? You know, that just kind of has cilia and goes around in pond water, right? Really simple, straightforward things. They're not very complicated, right? All the way up to say mammals like us, right? We have a huge range of behaviors. Depending on the type of mammal, you can find us all kinds of different environments, very adaptable, relatively complex, warm-blooded, right? So there's we have a lot more tricks up our sleeves, let's say, right, in terms of physiology. Right, we have, you know, fingers and arms and stuff, right, which worms don't and protists most certainly don't. We have a very complex developmental process, where we go from a single cell to this multi-million cell organism where different things do different jobs. They're very highly specialized, so on and so forth, right? And we have exactly the same number of genes as a soil nematode, give or take 1,000 or so, right? So there's a range of, you know, gene number in terms of eukaryotes, from the very simplest to the very complex, but there's no correlation between gene number and complexity, with a few exceptions, right? Just as there is no correlation between gene number, and this is what this diagram shows, between genome size and complexity, right? So they are very much not linked. You know, I'm not sure where wheat comes in, if it's a angiosperm or I think it's an angiosperm. Again, not much of a botanist. That's an enormous genome, right? It's at least 3 or 4 times the size of our genome, right? Absolutely massive. And it's wheat. It grows up, sways in the wind a little bit, gets mown down, gets turned into bread or tortillas, you know, and so its genome size is super large because of 2 rounds of genome duplication, right? So it has 6 sets of chromosomes for each, 6 sets for each chromosome, right? So there's really no correlation between what an organism looks like in terms of complexity or its behaviors and its genome size, right? That's really what the C value, which is the size of its genome, paradox is about, or enigma, depending on how you want to look at it. So yeah, but the only correlation that I can think of between organismal complexity and genetics is the number of chromosomes, right? So mammals down here typically have, I'd have to check some, but I don't know, let's say 15 or more chromosomes, right? stuff down this end, you know, you're looking in mid-single digits, right? 3 to 8, let's say, right? So that's really all I can think of in terms of a correlation, and really that's just reflecting, pardon me, evolutionary time, right? So the longer you've gone through evolutionary time from single-celled organisms to organisms like us, the more chromosome duplications have occurred to give you a large number of chromosomes, but you don't actually end up with more genes. You just end up with, you know, more or less the same genes, but now they're spread amongst many more chromosomes, basically.
Alrighty.
Cool. Awesome question. Does anyone have any more questions about this stuff? Because really the stuff that's totally new is what, slides 2 to 6, and then the rest of it you've already come across once already, so it should hopefully be somewhat familiar. It will also still be perfectly valid material for the exam, so don't skip over it thinking that you know it. Make sure you really do know it. This is a real cool picture. This was something that I was talking about in class. We didn't actually draw it up or do it in any detail, but this is what I was talking about, where every chromosome has its spot in the nucleus of a non-dividing cell, right? And so if you, this would be, let's say, one chromosome, chromosome 4, let's say, here. Then you got another chromosome over here, another chromosome over here, and so on and so forth, which I think is pretty neat. And then the only other thing that is new is this, which is a new, like an additional insight into what chromosomes look like at a genetic level, right? In terms of both gene density and also in terms of their chromatin. Does that all make sense? I'm going to assume it does.
Yes.
Okay. All right. So is everybody happy to go on to transposons? Or does anybody have any more questions about this? Totally fine if you do. We've got plenty of time. No. All right. So
I haven't done the transposon, so I won't be able to contribute anything to that discussion.
okay, that's all right.
So it's up to everyone else to ask questions.
All right. The flame of inquiry will pass on to other people. I'll just start picking on people. There are few enough people here. And I can actually see who leaves as well as a result, which is kind of funny. All right, so there's 7 of you, 8 of you in total. Funny. All right, so let's see. Let's kind of move this down a little bit. Okay, so and we don't have. We have 7 of you total, 8 including myself. Okay, so transposons are pretty cool, right? Actually, as long as you don't have background noise, you can unmute your audio and just ask questions as they come up, rather than kind of waiting for me to ask. There's enough of you that it's not going to cause any. There's few enough of you that it's not going to cause any kind of chaos. Okay, so and this will give you a little bit of head start, Michael, for going through this. I'm going to get as a quick aside, I'll be getting the next, what do we have next on the list? Pardon me. Oh, right, Rick. So we're getting onto gene expression and transcription. We're kind of playing catch up a little bit, to be honest. But I'm getting fully up to speed in terms of going through PowerPoints. So I'll be getting up the probably gene expression and transcription part 1 and part 2 will be up by the end of today. Right, so I really want to get a jump on this stuff so you have time to go over these things before these sessions. But I would strongly recommend you do, because the exams aren't going to be any easier. Right, they're going to be the same because, again, the big reason being this is a requirement for other courses which hopefully you'll get to take in person at the university. Hopefully this is not going to last forever. And so you need to kind of put in probably more effort doing this than you would do coming to class normally, right? Because you're going to have to take that burden of learning stuff on yourselves from those PowerPoints and audio files. So I'll get up the next 2 lots of lectures, right? So we can start getting caught up, which will be gene expression and transcription at least by today. So as best as you're able to, go through those, make notes, write down questions that you want to ask, right? You should ideally be coming to these sessions with a nice list of at least 6 or 7 questions, because if you've come across this for the first time, there's a good chance you're not going to understand it first time either, right? And that's what these sessions are really designed to do, so that in some senses, hopefully you'll actually do even better than you would if this was in class, right? Because this is now like an enforced office hours in a sense, right? If that makes sense. Okay, anyway, so let's get back to transposons. So transposons are cool. You might not realize this, but they are actually pretty neat. And they're pretty neat because they are independent mobile bits of DNA, right? So normally your genes, you get them from your parents, right? Or your alleles of each of those genes, right? You have a certain number of chromosomes, your genes are just there. That's it. Unless there happens to be a bit of a screw up during DNA replication or some kind of funky stuff going on where those genes get duplicated or, swapped around or something due to some kind of catastrophic event, they're there. They sit there, do nothing. They are static. Transposons, on the other hand, are pieces of DNA that can hop around, right, in that genome. And they're probably doing that inside you right now, right? If it's in your somatic cells, you keep that. If it's in your germ cells, then potentially your offspring could get a different genetic makeup to some extent than you have, right? Not just down to what genes you have, but whatever mischief, right, essentially that transposon has done. And they're able to do that by a couple of different things. One, they recognize a target sequence, right? So you see what I'm circling here on my screen. That's specific to that transposon, right? And when that's recognized, that DNA is cut, right? This is a double-stranded break. Each one of these is a strand, right? Top and bottom. And that transposon or transposable element gets stuck in there, right? It gets glued on. You remember how DNA ligase works, right? Where it makes a phosphodiester bond between phosphate and sugar. It does that, right? So it literally sticks itself here and here. So it's now part of that DNA, and those gaps are then filled in, which produce these things called flanking direct repeats. It's like a signature, essentially, of a transposon, right? If you look through your genome and you find these, that tells you that there's a transposon in between them. Oh, got a chat. Oh, I'm sorry. Can you see what's on my screen? I'm moving my cursor up and down. Bum, bum, bum.
I'm looking at slide 14, the first slideshow you did a recording for.
Oh, that's weird. Okay. Hang on, let's go back then. Yeah. Oh, okay. This is part of my learning how these things work. Right. So can you see slide 3 on the transposon PowerPoint now?
Yeah, I can see the transposon stuff now.
Can you see me move my cursor around?
Yes.
You can. Awesome. All right. So can you see, Michael, you're taking the lion's share of the load here. Can you see me circling the bit of DNA at the top right?
Yeah, I can see pretty much everything
Awesome. Okay.
that you're doing and seeing.
Super cool. Okay, that's awesome. Thank you for that, Rose and Michael. So, right, so this is that target sequence, right? So it's a very specific sequence. Doesn't have to be that long. There's one at least that I used to work on, which is, I think it's just AT, right? So there's even one there, right? You can see there's an AT sequence even in that little bit of DNA that was used. Or they can be quite long. It's very variable. But if that sequence is found, and typically it's found by an enzyme that the transposon produces itself, that DNA is cut. And then once it's cut, that transposon, right, which might have been cut out of somewhere else or may have been a new copy, gets stuck into that break. Right, so you remember how a DNA double-stranded break, right? You break both strands like that, and you want to put them back together again, like so, using DNA ligase, right, or some other repair mechanism. So what happens is that you've got your double-stranded break, and the transposon gets stuck in the middle, and then gets stuck onto it, essentially, right, using DNA ligase or an equivalent enzyme. So if you go down to where the speech bubble 2 is, you can see here that that transposon is now part of the DNA, right? So we've got the top strand here, which is what CGT, GAT, and then the transposon, and then you've got the bottom strand, and then the kind of the host DNA, right? Those gaps are then filled in, most likely by the host as part of a DNA repair mechanism. And that gives you these flanking direct repeats. Can you see those down at the bottom? They're in kind of purple, right? Those are a signature of the transposon. So if you look at it, essentially the direct repeat is that little bit here. See up at the top where the speech bubble 1 is, right? That sequence, as a result of the transposon going in, that sequence gets copied, right? So you go from 1, I've got to see where my webcam is, go from 1 to then 2. Those are the flanking direct repeats. And in the middle, between those two flanking direct repeats, is the transposon itself. So we're able to look at a genome, say our genome, and you can scan the genome using a computer tool and look for where do we have two short sequences that are repeated a relatively short distance apart, so 2,000 or 3,000 base pairs apart. Most likely, that's the signature of a transposon, right? And transposons make up, in our genome at least, almost half of our genome is made up of these things, right? That have been, just scooching around over our evolutionary history. Does that, any questions about transposons so far? They get more complicated, as you'd expect. Crickets? All right. So you're going to have to get used to this because this will be fun. Okay. So where are we at? An extra wrinkle is that quite often, typically actually, I mean it varies. Transposons are hugely varied things. There are these things called terminal inverted repeats, right? So if you put a where's my webcam? If you put a mirror in between, say where the B in transposable is, and you looked at that green sequence, it would be a mirror image, right? Well, I mean, kind of in like genetic terms. So don't try and do that. It's not going to work very well. But you get the idea, right? So these inverted repeats are the opposite of the other one on the other side, right? And these are, so if you look at the flank indirect repeats, those are like a signature of a transposon going in, right? It's a consequence of transposition. These inverted repeats are a tag that is used by the enzyme that does that to recognize where that transposon is. Right, so essentially it's a protein typically encoded in the center part of the transposon, right? So it encodes its own means to move. And that protein is transcribed, expressed, translated from that gene here within the transposon, wanders around, and then it comes back and it recognizes these inverted repeats. And that's what it uses to cut it out and stick it somewhere else. Right, so it encodes its own means of movement, which is pretty neat. It's also one reason as well why we have so much of this stuff kicking around, because quite often if you have a random mutation occur, let's say either in an inverted repeat or in the transposase itself that stops it from working, that transposon, if there is no other one around, you know, that can provide the transposase, that transposon is dead. It can't move anymore, so it's stuck there, right? And so you can see how over time, and we're talking millions of years here, you can accumulate an awful lot of these transposons, pardon me, both from them you know, being acquired and replicating, and also because they'll just end up, you know, kind of dying of old age, essentially, in evolutionary terms. Any questions? Nope. Couple of different types. Simply put, right, there are lots and lots and lots of different types of transposons, but a couple of types which are kind of broad definitions is you either have DNA transposons or you have what's called retrotransposons. DNA transposons are, they can be both replicative, right? So a copy is made to be put into a different location, or non-replicative, so that the existing transposon gets cut out and put somewhere else. In replicative transposon, you have an existing transposon, you make a copy, and that copy, go away. That copy is put somewhere else. So now you have two, right? And then obviously it can copy again, and you have four. So that's one way in which you can increase the amount of junk DNA, as they call it. There are also really funky ones called retrotransposons. These have an RNA intermediate. So that's probably best explained using this. So you'll see me on slide 6. You can also have this up open on your own computers too, if it helps. So here's a retrotransposon up here. Let's, where's the chat gone? Ah, come on. Oh, no, nothing's happened. It's, oh, I can't go back. If you have a chat, just stick it in again, or shout it out. It's totally fine. So this is DNA, obviously, because it's in our genome, and we have a DNA genome. But that DNA is then transcribed, much like mRNA is from our own genome, or ribosomal RNA, or whatever. That's transcribed into RNA, right? However, that RNA can't go back into our genome because obviously we have a DNA genome, or we have to make it back into DNA, and this is via a process called reverse transcription, right? So using an enzyme, as one would expect, called reverse transcriptase, and that takes RNA and uses that as a template to make a double-stranded DNA piece. So is that a question, Esmeralda? Or... No? Okay. What's next? So that's actually really interesting because that is the same process that viruses called retroviruses, use to insert themselves into your genome. So HIV, for example, is a classic retrovirus because it takes its RNA genome as it goes into your T cells, and that RNA gets turned into DNA, and that DNA actually goes into your genome. So that's the kind of crazy thing about some of these viruses, that even if you're not producing the virus itself, like you're not showing disease symptoms, it's still there. And if it gets reactivated, then you can have the disease again, right? Which is, I think, shingles might be another example of that. I know my nan used to have that really badly. Anyway, so, and then once that double-stranded DNA piece has been made, the same process applies for transposons in general. Cuts are made, transposon is inserted, those flanking bits of DNA are filled in, and then you have your flanking direct repeats. And oh, any questions so far about any of that?
Hi, it's Rose.
Hey, Rose.
I had a question about the replicative and transposition and the non-replicative transposition. So for the replicative transposition, is that where the direct repeats also get copied, or they don't get copied at all when they do that?
No, the direct repeats are not part of the transposon. They are a consequence of the transposon going into the DNA. So let's go back up to here. So we're on slide 3 again. And so the transposon is purely the bit in orange. Those flanking direct repeats are produced by transposition because of those staggered cuts that are made in the original DNA. Those staggered cuts, those single-stranded bits need to be filled in, right? So just by a transposon going into DNA, you get those flanking direct repeats. Now, if it's a non-replicative transposon, right? So I'm kind of getting to your question in a second. If it's non-replicative, when that orange bit is cut out and stuck somewhere else, those purple bits stay there, right? So you can see that just a transposon going in is going to be very disruptive, but even if that transposon leaves, you now have those two flanking direct repeats next to each other like so. Does that make sense?
Mm-hmm.
Yeah. So in replicative transposition, you have your flanking direct repeats, you have your transposon. It's only the transposon that's copied. I don't actually know how that's done for DNA transposons. I mean, retrotransposons, it's pretty straightforward, but for DNA transposons, I don't actually know how that's done. I've never worked with that particular type. But that bit of DNA transposon, that's what's copied. the whole thing stays there, right? So you don't take anything out, but you only copy the transposon itself, and then you stick that somewhere else. And in doing so, you generate 2 new flanking direct repeats at the site of the new insertion.
Oh, okay.
Yeah, so those flanking direct repeats are purely the result of something going in. When it gets taken out, they get left behind and they just get stuck back together again.
Oh, okay.
Yeah.
All right.
Yeah, so that's why I was saying that these flanking direct repeats are very much a signature, right? So if you're looking for these transposons, what you're really going to be looking for is not just the actual transposon itself, right? Because, yeah, for whatever reason, what you're looking for are these flanking direct repeats, because that's a classic signature of a transposon having gone into the host DNA.
Okay, thanks.
Cool. Yeah, they're a little bit weird to kind of think about, to be honest, because they're totally different to any other kind of, you know, genetics that we've really covered up till now, and they really don't play by the same rules as everything else that we've learned. They are really pretty funky. Okay, so as you can imagine, this can be bad for your genome, and it very much depends on where this happens as to how bad or not it is. So there's essentially 2 things that happen here. One, you've got this big chunk of DNA going in, the transposon itself. And 2, you have these flanking direct repeats that are created in the process, right? So given that most of our genome consists of, you know, these things, or other bits of DNA we don't really know what it does, then if this transposon jumped into, you know, some barren piece of chromosome 2 that doesn't have any genes in it, doesn't do anything, right? It may increase the amount of DNA, perhaps, if it's a replicative one, it, you know, may not. However, these often, for whatever reason, I don't actually understand why, these often go into, sorry, itchy nose, either coding regions or regulatory regions of genes. And so if this transposon was to go into a protein coding region, most likely that protein is not going to work, right? And that's actually the way in which transposons were discovered, right? So they were originally discovered by a lady called Barbara McClintock, who is working on corn genetics. And she actually noticed that several corn kernels were fleckled, right? They have, like you can probably remember from the lab exercise you did in genetics lab, they had like little bits of color in those corn kernels, right? And that's the result of a transposon jumping into that pigment gene and knocking it out. So it doesn't work anymore. It's also the reason for, if you, and I'm no botanist, I actually kind of enjoy gardening, but like the botany aspect, not so interested in. But if you look at, there's some very pretty variegated flowers, right? So these are flowers which have like really cool patterns of different colors around them. Those are almost always due to either a transposon or a retrovirus, right? Going in and randomly knocking out pigment genes. So if these transposon lands into a coding region of a gene, it's usually pretty bad, right, in terms of that gene function. Obviously, that's only going to be one of 2 copies because we're diploid. So it doesn't necessarily mean that that gene as a whole is going to stop functioning, just that that one copy of it will do. Obviously, it depends on what the other copy is as to what, you know, consequence that has. Also, if that transposon, right, this orange part here, if it jumps out again, right, if it's non-replicative and just kind of hops somewhere else, That's still going to cause problems because now you have an extra bit of DNA, one of these flanking direct repeats, left behind, and so it's still going to disrupt gene function. So that's if it goes into a protein coding gene. If it goes into a regulatory region, it's a lot more interesting, right? Because in terms of gene regulation, and I don't know, we should be getting to this later on in the course. You can both turn things on and turn things off, right? And that's done by different bits of DNA recognized by different what are called transcription factors, proteins that kind of bind to that DNA and help transcription or stop transcription. So if this transposon jumps into one of those sequences and, you know, stops it from functioning, knocks it out, so that protein doesn't recognize it anymore. Now you could turn off a gene in a particular region, or you can turn it on. And so quite often in evolution, one possible way in which you get novelty, like something new, a new trait or trait being expressed somewhere different or whatever, is because of transposons, right? They're very mutagenic, but they're random in how they do it. And so one of the theories in which how butterflies got their wing spots, you know how butterflies have those kind of, they look like eyes. They're used to scare away birds from eating them because it makes them look like bigger birds that might eat those birds. Those eye spots appeared several times in the evolution of butterflies, you know, I think like 5 to 10 million years ago, give or take. And one of the possible ways in which that could have happened is if that transposon jumped into a bit of regulatory DNA and essentially turned on pigment production somewhere where it shouldn't normally be turned on, right? So these transposons are also an interesting way of potentially gaining something which you wouldn't have had before. Still via mutagenesis, right? They're still doing damage, but the damage that they do can vary, and it can vary in their consequences. Another thing, quickly just to wrap up, is that transposons also, because particularly replicative transposons, if you think about it, you've got, let's say, chromosome 1 here, right? It's, you know, chromosome 1. It's very different from chromosome 2 over here. And so these two are never going to swap DNA, right, in meiosis, let's say, or repair and double-stranded break. And they don't swap DNA because they're not the same, right? They're very different. And so there's no matching that can happen during prophase or during DNA repair, which would stick them together in any way, right? Now you can imagine if you have a transposon here, which copies itself and sticks in that copy over here, now you have sites of homology, sites that are the same in different chromosomes. And so now you can imagine how during potentially during prophase, or more likely during double-stranded break repair, those chromosomes are going to recombine, and that's going to generate a completely different, two different chromosomes, or maybe even in the case of chimpanzees, my fingers are not doing their job here. In the case of chimpanzees, you could actually get one larger chromosome as a result from two smaller ones, which by accident kind of join together. So they can have some pretty major major changes as well. Does anybody have any questions about transposons? Don't worry if you don't. Write those down once you go over this again and bring them to the next session, right? Each session is not on a specific set of lectures. It can be on really whatever you want to ask questions about.
It's Rose again. I had one question. it was, where do these transposons like originate from? Like, are they just in our DNA? Like, it's just there, and then it just come about?
Oh boy, um, that is an astonishingly good question. And
Sorry.
no, not at all. I've thought about this before. I don't know, and I'm not sure if it is known.
Mhm.
I've certainly not come across any answer that I can remember. I studied these at, many moons ago when I was in New Zealand and Australia. So that's like 12 years ago, 14 years ago. So maybe something has been found in the meantime. My guess would be they're relics of retroviruses
Oh, okay.
or um, some kind of no, actually, it doesn't have to be retroviruses, but retroviruses are quite often ones which go into DNA. My guess would be some old virus or of some sort which integrated into DNA but kept the means by which it did so.
Oh, okay.
Right?
So
Because
it's like the transposons, like essentially they were RNA that got changed into DNA
yeah,
or
so
transcribed.
that's how retroviruses work. And my guess is that, I mean, and also DNA viruses can, hello doggo, my doggo
Oh,
came down to say hello. She's such a teenage dog. She's only just got up. So DNA viruses can also integrate into your genome, and that's often how you can have viral diseases which kind of go away and then come back, like in times of stress and stuff like that, or changed immunity. They kind of pop back out of your genome essentially. And so my guess would be that the transposons are essentially ancient relics of old viruses, which kind of got in somehow, but kept a very like stripped down version because they are very minimal. There's not very much to them at all. They're much simpler than viruses, for example. Kept the means by which they can move around. That would be my guess, but I don't actually know, to be honest.
oh, okay.
Yeah, good question though. Cool. Does anybody have any questions? Again, no worries if you don't. Keep those questions for Tuesday. Not Tuesday. This is Tuesday. For next Thursday, and I'll be more than happy to answer them there as well. Cool. All right. So if you're all good, I'm going to call it a day. Going to have myself a cup of coffee and get ready preparing the next few lectures so you can go through those and get prepared ahead of time. Okay.
Thank you, Dr. Crook.
Thank you, everybody. Great to hear or see of you again. Hope you're all staying safe. And we'll see you soon. Where we go? Here we are.